Hello and good evening to this very first international conference on upper airway solutions in COVID-19. I have a tremendous group of people with me from all over the world, and I just wanted to take the opportunity to thank them for being here. I'm going to give a quick introduction to the people who are here, both our speakers as well as our panel. I'll hear a quick word from our sponsor before we then move into the bulk of the meeting. And so with great pleasure, I'd like to bring in first, we have Stan Lech. Stan, where are you?
Hello, I'm in Los Angeles at the moment, but I live in Miami.
Excellent. And we also have?
Hello, I'm Debbie Black, and I'm in Gold Coast, Australia.
Wonderful. And?
Marco Sanchez. I am in Miami, Florida, US.
Great. And as well, Vanya, can you hear me?
Yes, Vanya Loroch. Yes, I can hear you just fine.
Wonderful.
Can you?
Yes. So Vanya is from Switzerland. He's another one of our speakers. And our panel here, we'll quickly introduce. We have John.
John Abilis. I'm from Florida, and I am originally from Rhodesia.
Excellent. And we have here Jennifer.
Aloha, I'm Jennifer Smith, and I'm currently in Hawaii.
Wonderful. And finally,
hi, I'm Chris Newton. I'm from Yorkshire, UK.
Wonderful. And we also have helping us in the background as well to coordinate some of these things here. You may as well say a quick hello, Jackie.
Hi, I'm Jackie Allison. I'm in Toronto, Canada.
Wonderful. So this is truly an international panel of people, and this should be quite interesting. Our format is we're going to have each speaker speak for 15 minutes, which would include questions as well. And we're going to try and be very sharp in our transitions. At the end of the four speakers, we will then have a tiny short interlude where we'll speak about the sponsor, and then we'll go into the panel discussion where the panelists will be able to discuss generally some of the ideas and speak directly with each of the presenters. Does that sound about okay to everybody?
Excellent.
That's great.
Excellent. So what I'll do is that we will just hear a quick word from our sponsor this evening.
Macmillan Research is one of the Vejon Health group of companies, which comprises health and media businesses. Macmillan Research, along with Dementia Crowd Research Limited and VibroScience Limited, focus on innovative medical research to find solutions for a selection of common diseases as well as strategies to manage them. At Macmillan Research, we take a unique approach to medical research, applying the fundamentals of physiology and pathology to understand disease. Through detailed research on COVID-19, a theory of autoimmunity to angiotensin-converting enzyme 2, also known as ACE2, was formulated in April 2020. At that time, the hypothesis that COVID-19 is a viral-mediated autoimmune disease was not regarded by many as plausible. Over the past year, the theory of autoimmunity in COVID-19 has gradually gained credence. Dr. Macmillan, Executive Director of Macmillan Research, collaborated with experts to publish scientific papers in May 2020, explaining the concept of autoimmunity in COVID-19, and March 2021, proposing a more detailed and comprehensive explanation of the symptoms associated with COVID-19. The principle of autoimmunity in COVID-19 is that the viral spike protein binds to serum ACE2 to trigger an autoantibody response. This may lead to severe disease in people who have elevated levels of serum ACE2, including the elderly, and those with obesity, hypertension, heart disease, and diabetes. Sex and ancestry are also determinants of elevated serum ACE2. This theory of autoimmunity could explain the symptoms, timeline and associated comorbidities in people with severe COVID-19. A COVID-19 research project has been designed with the aim of advancing our understanding of immune dysregulation in COVID-19. This is the primary cause of lung damage which can lead to death. The outcome of the research project has the potential to change the course of the pandemic.
And thank you very much for that. And what we will do is, again, we're going to do a very simple pattern with regards to our presentations. We're going to start with Marcus from the USA, followed by Debbie, followed by Vanya, and then Stan, assuming that we don't have any technical issues along the way. So we're going to probably go straight into our presentations at this point. And we will probably start with Marcus, and we'll allow him to share his presentation, and I'll ask him a few questions at the end just to get a better clarity as to what's going on. All of our speakers and presenters are going to be discussing with our panelists in the next hour when we come to our panel discussion. I hope that makes sense to everyone now. So let's get started and welcome our first speaker. Hi, Marcus. How are you?
Excellent, Dr. Macmillan. Thank you very much for having me here. Very
Wonderful.
excited. Very excited
Yes.
to share.
So just get straight into introduce yourself, and then let me know when you want me to bring up your screen.
Absolutely. Marco Sanchez-Gonzalez. I'm the Chief Scientific Officer of Dr. Ferrer BioPharma, based here in Miami, Florida. I'm a translational physiologist, and one of the goals of our agenda is to take molecules that have potential therapeutic capabilities, to take them from the molecule all the way to the communities. As a matter of introduction, throughout this pandemic, we have been seeing different kind of strategies that we are mounting in our fight against the pandemic and the virus. We talk about using masks, we talk about vaccines, treatments, drugs, but one of the strategies that usually are overlooked are the strategies that have to do with the nasal oral hygiene and the upper respiratory airway, which is the portal entry of the virus. So I would like to kickstart things off with that. And as we say, the best for the last, that's why I'm first today. Please, let's start with the presentation. As a matter of background, just a very quick in respiratory physiology. Remember, the pulmonary system is one of the systems that takes longer to mature. That's why we are very susceptible to diseases of the respiratory system. So they go across the age range in people. So there's a specific aspect of the pulmonary system that we call the nasal cycles. And I'm going to go into briefly into the physiology and why this is important in the context of respiratory disorders, and in this case, COVID-19. Also taking that into consideration, how inhalation, in this case specifically, intranasal therapies could be one special or added advantage over other kind of therapies in our fight against COVID. So the nasal cycle basically is a series of cycles in which the flows in one part of the nose fluctuate with the flow on the other part of the nose. Some of the physiological advantages of having these cycles is that the flows and the cilium that has the capability of removing dust, removing particles, including pathogenic particles, are constantly protecting us. So there's an asymmetry. There's a fluctuation that involves the autonomic nervous system. Yes, it has some circadian rhythm associated to it. But the most important thing is, among other functions, we have the production of naturally nitric oxide, congestion with air going in and out, the sinuses, the mixture. That nitric oxide serves various purposes. One important purpose is serves as a microbial or antimicrobial agent, antiviral agent, but also provides the fuel of those cilium cells. So in principle, if we keep these nasal circles healthy, now we start to stop any potential disease that we have. So that's the first barrier in terms of disease prevention. In the same token, the nasal and inhalation delivery has been gaining popularity after in between the 50s and the 70s, everything was about systemic drugs. Now we have been recognizing that the nasal route of drug delivery provides certain advantages that the systemic, or in this case, a consume or gastrointestinal route doesn't really offer. Why this is important? This is important because still we have a very important amount of people suffering of upper respiratory tract infections and respiratory diseases, period. Then you need to also consider that the portal of entry of COVID is the nose. But we have not been keeping enough attention to that important fact. Respiratory diseases, in this case, for instance, rhinitis, it's so common that sometimes we think it's normal, which is not. All of these diseases that are related to the respiratory system, they have an important impact in our lives, in their economy. But more importantly is that many of these disorders are associated to poor respiratory cycles, specifically the nasal cycles that we spoke before. And COVID is not the exception. COVID is not the exception into those. These are very recent data that we have here. But when we start thinking about what will be a good candidate molecule, what candidate molecules can help us to treat the disease even before it starts, that we can prevent, or if for some reason you get infected, how we can stop the virus on its tracks. So that allows us to start looking into those candidate molecules, not necessarily novel molecules, but we can look at existing or pre-existing molecules that are out there, reformulating them in a way that target the nose and using the devices to basically deliver the drug in the point that we want it delivered. In this case, for the most part for COVID, we're talking the nose and the nasopharynx. So from those three molecules that I would like to talk about, some of these are medications like chlorpheniramine. It's a antihistamine. It's been in the market for over 40, 50 years that has anti-allergic, anti-inflammatory, but interestingly, antiviral properties. Xylitol has microbiome and sanitizing kind of properties. And then you have grapefruit seed extract, which is a natural compound, but has multiple broad spectrum microbial or microcidal actions. So how we can combine these molecules, or what are the advantages of using these molecules in the nose? It's part of the drug development question that we ask ourselves. So interestingly, we look at what is the potential antiviral potential of some of these components, and we look at the grapefruit seed extract xylitol as an antiviral, basically a component. Here I have a viral cell that was cultivated in a media rich in xylitol. Okay? And in these experiments, we were able to demonstrate that for some reason, xylitol is blocking the entry of the virus inside the cells. Now, if we use a kind of a blanket of xylitol in the mucosal, potentially at the translational level, we can suggest that potentially that can serve as a first barrier to the entry of the virus into the cells. Also, we tested the similar experiments using chlorpheniramine, and the results were basically the same. The idea came from prior experiments looking at the potential of chlorpheniramine for the treatment or as an antiviral molecule against influenza A and B. So potentially what is happening is that you have the cell receptor, you have the virus, and these molecules that we're talking today, chlorpheniramine, one of them, basically are in the middle of that sandwich. So when that happens, you are basically blocking the entry of the virus into the cell in a very effective fashion. We were able to show a 99.7% of all the viral particles that were blocked by this mechanism. That also took us into the consideration of the advantages, both as a prophylactic or prophylaxis treatment, as well as a treatment for viral disease, and in this case, a COVID. Thinking about the expression of ACE2 receptors, they're very high in the nose, very high in the nasopharynx. That's why the virus take that as a preferential site of infection. If we use substances intranasally that can block that, chances are that we're going to decrease the chances of the virus invading and spreading from the nose nasopharynx into the other more vulnerable tissues. We also thought about how we can distribute this drug in a uniform manner and how we can target the place that you have the most accumulation of the virus. So the angles of basically penetration of the atomizers is very important to consider. We even developed a good nozzle that penetrates and it's used as a blanket to basically cover all the mucosal. We also look into this in cases of patients, and at the clinical level, we have been testing it against COVID. We also have been doing some observational research to see people using these substances in a daily basis, more as a hygiene, more in case of a hygiene or a nose hygiene, as opposed to more with the medication for the intervention. And the result has been very, very promising. Observational studies suggest that people in these kind of strategies, they don't get as infected as people that they don't follow these kind of nasal hygiene, upper respiratory airway strategies. And that's all from my end.
Wonderful, wonderful, Marcus. That's tremendous stuff, because it looks as though this is a strategy that has real world significance. I'm assuming this is not just relevant for COVID-19. You guys are looking at this for other viral infections, I assume.
Correct. And evolutionarily, upper respiratory viruses has been with us forever, and still we have flu, we have multiple coronaviruses, not only the SARS-CoV-2, over 200 species of common cold. So looking into nasal hygiene, looking into specific medications to control the viruses in the place where they start has been the main focus and strategy behind our research agenda. At the same time, we look into the formulation, We look into the distribution and specific instructions on how the patients should use the nasal sprays. It has inserted in a certain way, the angles of penetration, all of that plays together into a more efficacious or effective treatment.
And would you think that it would become almost as standard for people when they are, it's in the winter months? Even if we take COVID out of the picture, they would use this every day or once, twice a day? How would that work?
Same as we do oral hygiene and we brush our teeth at least twice a day, we are recommending the same. You put these nasal sanitizers next to your toothbrush, you wash your mouth, you wash your nose. It is important to mention here that this is not wash your nose strategy. This is a hygiene, and we have to be careful with the kind of substances. We don't want to kill all the microbiome. That's a different discussion. It has to be gentle enough to kill, let's say, the bad pathogens, and has to be good enough to keep the good bacteria and good viruses in your nose, because they're part of that barrier. And that's a different topic, but it seems to be the future of therapeutics these days.
And this will be a cost-effective strategy, I guess. It wouldn't be too expensive for people.
That's correct. That's the other thing, is that we're trying to, by repurposing existing molecules, there's a good savings in terms of development, that at the end we want to have this right there over the counter that people can utilize in an everyday fashion. And as an additional strategy, this is not the cure of all, there's no silver bullet in medicine, as we know. This is an additional strategy to the existing arsenal that we have against viruses like COVID.
Wonderful, wonderful. Well, thank you very much, Marcus, and we will discuss it in a little bit more detail when we speak with our panelists afterwards. And so moving straight along into our next speaker, all the way from Australia, we have Debbie Black, and I'll let her introduce herself. Hi, Debbie.
Thank
Wonderful
you,
to have you. Yes, and so tell us a little bit about yourself and your company.
Sure. I'm the chairman of Bionic Air, and Bionic Air represents a medical device. This is what it looks like, and it's invented by Joshua Shaw in 1967. But it really is not very well known globally, but is very well known here in Australia. So unfortunately, Joshua passed away in 2019, so I'm honoured to take forward his life's work. And this particular device has very good results in Australia on colds and flu, especially, with 95% of our patients reporting that they just don't suffer severe colds and flu any longer, if at all. So to have a device that's 53 years old and with over 1 million patients in Australia, we've had no recalls, no adverse events in that time. And the medical device is very, very easy to use. It sits on the bedside table, and there's only one button for the consumer to turn it on. There's 4 needles around here. There's no motor in it. There's no chemicals. There's no filter. And it's actually a type of cold air plasma. And so what I'd like to do today is introduce you to cold air plasma, and also to introduce you to some of the results we've achieved. So Phil, if you wouldn't mind putting on my slides, please.
Wonderful.
Okay, great. Thank you. So first of all, I will be, because we're talking to a professional audience, I will be providing some results we've received in humans that are not necessarily on our medical claims at this point in time, and for research purposes, of course. So first of all, what is plasma? Plasma is a type of matter. So we have solids and liquids and gases, and plasma is the smallest. To consider plasma to the human eye, if you think about the aurora up here, that you can see the beautiful green lights above the Arctic Ocean, and that is plasma caused from electron showers from the sun and the magnetic fields of the earth. So as you can see here, the sun is a type of corona, and that's plasma in the middle, and that's what we produce here. Oops, just went too far. So there you go. I don't have time to go through this today. This is a very rough summary of what's involved in this technology. It is quite advanced. And the way we produce cold air plasma species, they're reactive oxygen and nitrogen and oxygen singlet, for example. We do this in negative and neutral polarity through pulsed electromagnetic fields and novel portals that we use into and out of the human body. We do this using the respiratory system, and we also do that using the skin. And the device is a systemic selective adaptogen, which means it works across all systems of the body. It works in a selective way. It is able to eliminate pathogens that should not be there, whilst it has the ability to protect the human cells at the same time. It works as an adaptogen, which means that each individual body is getting the dose that they actually require for that particular body, using the body's own systems to measure that. So I wanted to talk a little bit about the transdermal. Here we had 12 months before I was in Wuhan on the front line, we had a breast cancer case, and 4 weeks prior to the lady having surgery, the report came back from pathology showing us that the tumour had no cancer cells left in it, surrounding tissue had no cancer cells, and the lymph node had no cancer cells. We treated this lady transdermally, and it's the first case we had of being able to reach a result confirmed by surgery and pathology of a 5 centimetre deep tumour. This is an example of a diabetic foot, once again, showing the antibacterial and controlling of redox solutions of this device. This particular gentleman was to have his foot amputated, and within 3 months we retained it and he's walking fine. And a basal cell carcinoma, same story. Within about 3 weeks, you can see using a transdermal approach that this particular patient also performed quite well. So here's some photos of in Wuhan, where we used the device in both respiratory and transdermal treatments on compassionate grounds. So looking at the size of the species, when you look at COVID-19, that big purple circle you can see there is what the size of COVID-19 would be inside an aerosol. And this little tiny dot you can see there, well, that's a gas particle, and we're talking about electrons and plasma, which is much, much smaller. So we're able to produce a huge amount, millions and millions amount in comparison to one virus. So when COVID-19 started, one of the, and also we're not registered to be treating COVID-19. It's not on our medical registration here in Australia, but we do treat viruses and pathogens and bacteria of the body and blood, and that is on our certificate. So we did need to make sure that we could protect our own patients. So we started looking into what we could do. We didn't have any idea about sort of frequencies and settings and strength that we would need to do to using COVID-19. So I did quite a lot of research and ended up going back into Pythagoras and Plato days and into geometry, maths and astrology and numerology to come up with the triangles of the icosahedral inside the COVID-19 virus, because I knew we could make a difference to the matrix, the lipids, by using our mechanical vibrations of the virus. And then if the resonant frequencies can, as you know, resonant frequencies can make a difference to a building falling over or a bridge being demolished. So surely it'd be able to do the same with the virus. And we found that we were able to get success in this by using this methodology. And what it did was it gave me a sound. So at the end of the day, the 20 triangles using the Pythagoras factor 9 grid meant that I was able to produce a perfect A-sharp sound. So I was able to turn the COVID-19 into an audible sound that I could then do all this work back to front using my device to then try and work out what frequency and what strength we could do to resonate with the COVID-19 and basically blast it. And that's what we do. And so when we actually believe it's the same kind of thing we're doing with other viruses as well. So these mechanical vibrations actually inactivate the virus. it's a very different approach to everything else. So how do we transport that energy inside the body? And you would think that that water doesn't have a memory, but it does, and we've been able to show that. And you can see the work from Professor Loroch in America. He's done some great work in this area to show that the spinning species, when the cold air plasma puts it into the respiratory system, is actually spinning at around almost 2000 Celsius. So it's very, very hot. It's spinning really, really fast and vibrating and shaking pathogens to comply with what it wants to do. So as you can see here, the water actually holds hands around the frequencies. So those little blue things is a water molecule. You can see the two hydrogen, the little white circles, and you can see the energy being encapsulated inside. And we believe it stays like that into the body until the resonance is able to communicate with where it's heading. So in our business, we looked at how many of the respiratory colonies could we eliminate in the first 3 hours of treatment, remembering that our patients use this machine every night as they sleep. They put it on their bedside table, they turn it on when they go to bed, and they turn it off when they wake up. So they usually have 7 to 8 hours exposure. And here we got an average of 72% reduction in the first 3 hours. This was when I came back to Australia after being in Wuhan. A surgeon called me to let me know that he's got a 23-year-old boy with glioblastoma, which had metastasized, and he was in palliative care. And he asked me if I would go and see him and assist him with his blood oxygen level. It was currently at about 84 to 88, and we were able to increase that to 95 to 96. And the gentleman only had half a lung, so it did take 36 hours for it to work. And the good news is he's currently in remission 12 months later. COPD is another area, and sleep apnea. Once again, we're not registered for this. We don't advertise to treat this condition at all, but a lot of our patients are experiencing this. And we had one patient call us and say that he felt better using the Mark II than he did using his mask, which was very strange. We decided to investigate it, and we found his blood oxygen level fell by 12.72% on the mask and increased by 34% using the Mark II without a mask. We've since increased that to about another 40 people in a little trial we're doing, and we're getting the same results. So we don't understand why that's happening yet. Asthma and emphysema, we've been treating those for 53 years, along with all other respiratory illnesses. And as you can see, the most common patients say that they don't need to use their puffer as much, and same with emphysema. Once again, this is just our customers buy our device so that they can breathe better, and then we follow them through and we find out what conditions they have, and we keep records of all that information. UNSW did a report found on 973 patients in 2009 and found 95% reporting fewer colds and flu. We started another one in 2000 and we've got 144 people, and so far have 97% fewer colds and flu. This is from the late Professor Singer in the mid-1980s, and he found that the immune system reactivity had a convincing probability of 0.001, and spore precipitation was statistically significant as well. This was when we were looking into the immune system, what's really happening. We decided to test live blood culture, and we looked at phagocytes. And you can see after 30 seconds exposure in vitro, we were able to see the germs being actually eaten up by the phagocyte. And this is one illustration of how what we produce is stable in the body's water. And here's another one to show that it's a stable detox regulator between the external ROS system and the internal ROS system through the receptors in the lungs and the neutrophils and the leukocytes. It's really quite a complex technology. And this one here shows how we infiltrate the rest of the body. So when the species is absorbed through the nose and through the mouth, it impacts the vagus nerve at the back of the nose and the back of the throat. And from the vagus nerve, we have the ability to cross the blood-brain barrier because our species is so small. And when we do that, we reach the higher regions of the brain and affect the central nervous system. And then that obviously talks with the sympathetic outflow of the heart. So we know this because we're seeing results in nervous and hormonal regulation of our patients. And also it's one of the ways that we've been able to test, of course, when patients are asleep, their serotonin levels drop and they're in their blood. And as a result, their melatonin levels increase to come around as an antioxidant in the body. And here's some most recent academic pictures that I've sourced. So it shows the plasma phase, the gas liquid phase, and then the liquid phase of the different chemical changes that happen between the particles, and also what's happening with the oxygen singlet mechanism. And so it's also really important to remember that there's also, when I talk about the resonance and the oscillation, is the whole issue of quantum tunneling, because non-linear coupling between the dipole oscillators in the cold air plasma caused thermal environment, it channels the energy into a single coherent oscillator, and then that resonates and through the fluids and the water channels and the currents of the fluids of the human body. And that then activates the cells at the biological temperature. So the body in this environment is able to activate and store energy to control its own chemical reactions as it needs for the day. And that's what we do. This device is not a device that's I'm saying is going to heal this and heal that. What it does is it puts the body back into the position it should have been in the beginning, or was in the beginning of time. And unfortunately, we've been removed from our environment, and the body's natural state has been altered. So we have the ability to bring it back to its natural state, which is why drug companies and vaccines tend to get better responses when their patients start using this technology as well. And that's enough for me because I'll run out of time.
Wow, yes, you probably have. Just before our time is completely gone. Oh, sorry, should I put it back up? Do you need it back up?
Mm-hmm.
Oh, sorry about that, Debbie. Can you hear me, Debbie? Can you hear me?
Sorry, can't hear you.
Oh, sorry. Can you hear me? Are you hearing
sorry,
me now?
Phil, it was me. Sorry,
okay, yes. So I just wanted to quickly, what were you doing in Wuhan with regards to COVID? If you can answer that in 10 seconds.
We treated about 200 patients on the front line. I was there on compassionate grounds helping people suffering from COVID-19.
Wow. Well done. Well, what we'll do is we'll get back to discussing this in a little bit more detail. So thank you very much, Debbie, and we'll talk with you and the panelists in a little bit. We're so sorry that we've lost Vanya. Vanya is from Switzerland and has been, we just haven't got enough connection for him to share his work. But his work was primarily around using technology with nasal vaccination. And that's a very important part that I think that I'd hope that Vanya would present, and it would add another dimension to the panel discussion. But I think we will still be able to discuss it with some of the panelists, even though we didn't have Vanya presenting. So moving straight along, we're then going to introduce Stan Lech from the US to have his chat with us. Hi, Stan. How are you?
Hello, Philip. How are you?
Wonderful, wonderful. So I'll let you talk. And the only thing you haven't shared with me, Stan, is your screen. But I think you can keep on talking and share where you are, what you're doing, and your company.
Absolutely. I will share that right now with you, sir. Mm-hmm. Yes. So I'm Stan Lech. I'm currently based in Los Angeles. I have a company called Immune Mist, which is only a fairly new company, but we're looking to target, and exactly what Marco said, really set up quite well, was the importance of the nose and the mouth in COVID infection as well as transmission. And so it's just a start. We're starting it up, and we'll see what happens. Prior to that, I was involved in 3 other startups, one in the US, one actually in China, where I lived for a number of years, and one that still goes on between US and Sweden. So I'm a bit of an entrepreneur. And then prior to that, I was the head of Sanofi R&D based in Paris for the consumer healthcare division. Then prior to that, head of innovation for GlaxoSmithKline which is a great company as well. And so I've done both corporate and now entrepreneurial stuff.
Wow. Wow. All right. Do you want me to share your screen?
Please, sir. Yes. We'll see how this works out, because I know there's been a little bit of a trick here with my side for some reason. And let me just see here. Start from beginning. Can you see that?
Yeah, we're seeing it okay. Yeah,
okay,
it's
good.
fine. Just go ahead with it.
Perfect. So, one of the things as a product developer and someone that looks at things maybe very simply and not overly complicated is it seems we've forgotten a lot of good therapies that we knew for, literally 100 years. And here it's nasal and oral hygiene. And Marco set it up really brilliantly. And that this is something I think we need to really look at. Now, on the first here, the basics is, the reality is that, with all these other sexy treatments being done in pharma, high cost stuff, also vaccines, a lot of the simple treatments, fundamental treatments have basically gone by the wayside. And so my point is to encourage the researchers that may be listening here to really start looking at some of these other products that are has high efficacy and certainly high safety, because they're both affordable and very easy to use from a consumer standpoint. Now, if you look at the basics, 70,000 liters of air come into your nose and mouth every day. So your nose is a very large vacuum cleaner. And it's no doubt that you will collect things in your nose and mouth that you don't want. And clearly the nose and the mouth are the gatekeepers of the lungs. Now, we all have been told, how COVID is spread from person to person. It's infected droplets that are either coughed or sneezed, potentially even touching our hands to our eyes and so on. But most of the infections start in the nasal pharynx, oropharynx, nasal cavity. And eventually these virion particles find their way to the nasal lining. They get through the mucus and the other barriers that we put up as a human being. And they start to infect the host. It may take several days for this to happen. Some literature says 3 to 14 days. Others say, 5 days on average. So the point here is, once these virion particles start to multiply and get to very high levels of viral load, you're either going to go one or two ways. You're either going to become infected and symptomatic, or you're going to be asymptomatic and potentially a spreader. And so, the point here is, can we intercept the virus early, particularly in the nasal and oral cavity, before people can spread it or before it can enter their lungs? That's the basic premise. I was going to talk a little bit about previously other studies have been done on hypertonic saline and surfactants, and they're all interesting too, and some of the results are quite remarkable. And so anybody interested in hypertonic saline and other therapies, there are research articles out of Vanderbilt, and there's one out of Elvis. It's called Elvis Study, that is from Edinburgh University. But what I want to talk about here really is the summation of where I ended up, where therapy probably will be the best. And this is using rapid-acting, broad-spectrum antivirals, which are at low concentrations but have incredibly high impact on the virus. Here we're talking about 4 and 5 log reduction in vitro and in vivo of the COVID virus, and at very dilute concentrations. And so povidone iodine, or PVP iodine as it's known, has incredibly good safety and tolerance up to about 2.5% weight volume. And then after that, you can run into some irritation. But PVP iodine has been used for over 60 years, both in RX, OTC, and also surgical use. And in fact, a shout out to Jesse, who is the CEO of Halodyne. He's an ophthalmologist. I mean, he's been using PVP iodine in surgery for the eye, in particular, to stop viral infections. So, it's really kind of commonly known among experts, but it's not broadly known that this molecule can be really that effective. The great thing is it's very cheap. You're talking about $1 to $2 per day, which really has a big impact. And funny enough, when you look at the clinicals, which I'll show in a minute, the reason why a lot of the clinical data is generated in India, Bangladesh, and other parts of the world is because A, they have no vaccine. B, cost is a big issue, and PVP iodine addresses a lot of these issues. So let's go and look at some of the clinical studies and top-line analysis from a really great website that exists is called C19pvpi.com, but they have other therapeutics also analyzed. So let's look at PVP iodine here and outcomes. These are clinical studies. Now, as I said, unfortunately, it's a bit of an orphan category, these nasal therapies, when it comes to kind of investigation and research and investment. But for the ones that have been done, Chowdhury shows some very interesting results out of Bangladesh with 606 patients. Here you're looking at an 88% reduction in death in that study, a 86% reduction in hospitalization, and across the board reduction in viral load. Probably not by coincidence. Arafin, recent study, it's only a couple weeks old actually, looked at 3 different formulations of povidone iodine, 3 different concentrations.
Stan, just to
Yes.
say, if you can move the slides along, because we are just seeing
Sure.
the main slide. Yeah.
Oh, can you see it now? I'm moving it.
I would suggest that, yes, there you go. And now we can see just, yeah. Mm-hmm.
Got locked up, I think, from, again, my side. So this is the study I was just talking about. So you have Chowdhury showing reduction in death, reduction in hospitalization, reduction in viral load. Then Arafin is only a couple weeks old, actually. This is several different formulations of povidone iodine, either used intranasally as an irrigation or as a spray. There was a good dose response. Nasal irrigation seems to be the better choice, with higher levels, about 0.5%, I think, or 0.6% being the best. And then this other folks have run a study called SEAT, which again is a fairly recent study in healthy young males in a dormitory setting where obviously they want people to get infected. You're looking at hydroxychloroquine or povidone oral spray. And here, you had a reduction in the severe cases among those healthy individuals. And you also had a total number of reduced cases. So while this isn't the world's best data, I think it merits a further look, especially, the folks we have on the line here. I think this is really promising. When you look at povidone iodine as a therapeutic, compared to other therapeutics, realizing this is the only topical therapeutic, it ranks right up there. All right, granted, it's 5 studies, ivermectin's 58, but I think it's giving us a signal that there's something here. All right, so I'm going to jump to my last slide because I knew we had a chance of running long, so I wanted to run a bit short. So given what we know in the data already about povidone iodine, given its risk-benefit profile, given its low cost, given its ease of use, nasal sprays and oral sprays tend to have high compliance as well. I believe, certainly, and my colleagues believe, that COVID can be intercepted in the nose, in the infection cycle, but also in the transmission cycle. Now, larger clinical studies are needed, and I would really beg that people who see this, who have the resources to do it, should do it. But we also need to recognize that this is an immediate tool that can be used right now because there's products on the market. And there's a low chance of these things having contraindications, and they can be used in combination. The last thing really is, the general safety is quite good, but, pregnancy and people with thyroid disease probably would need to have a bit of a watch out. And then lastly here, with something that has broad spectrum capability that is very safe, can we use it in targeted ways to disinfect various people, various populations, or just, various situations where we can either slow or stop the spread of new variants. So with that, I'm done.
Wonderful. Excellent. Excellent. That's really interesting, Stan. And do you actually believe that povidone iodine can become the lead ingredient in the battle for COVID-19?
Well, I think it has a very good shot. And the reason why is that it's been used for 60 years. And it's very well respected as an antiviral, antibacterial. There is no chance, I believe, of resistance because it's a mechanical action on the virus. So, using it would not lead to further complications, I don't believe. So I think based on that, the safety, tolerance, and efficacy, I have not found an ingredient yet that has this pedigree.
Wow. And so who would best benefit from using something like this?
Well, I have a few folks in the family that, police officers and other people that have high exposure, so to speak, right, to the populations. And so I think really, starting with people that have high exposure, maybe people that do not want to have a vaccine, that there are people that don't want to be vaccinated. Certainly that could be good. But I do see this as a broad approach. I think this could be used, in young adults, maybe to stop any transmission to the older adults. And I think it can be used prophylactically if there's someone sick in the house. So why not use it so you don't catch something? And then lastly, in high-risk populations, potentially nursing homes and others, where you may get some breakthrough or may get some other things happening.
And so practically, how often would it need to be used? Is it a once daily, three times daily?
Most of the protocols that in these studies have it being used several times a day, but these are actually in treatment, right? Treatment protocols mostly, except for obviously the one study where it was used prophylactically in the healthy young adults. So my point here is if you have COVID and you want to reduce viral loads, I think 3 times a day is probably normal, probably for 14 days. And then prophylactically, I think, this could be situational. If you're high exposure, then maybe use it, a couple times a week. I do take Marcos's point around, you don't want to screw up necessarily the nasal biome, but in general, they recover, and the safety data at the moment shows that these products can be used, upwards of 80 days in a row. So, relatively long-term use.
Wow. And is it affordable?
About $1 a day.
$1 a day. Wow. That is tremendous. Well, great stuff. Well, thank you very much again, Stan. And again, what we will be doing is that we will be discussing all of these ideas with the panelists in a short period of time. We're a little bit contracted in time because we've lost Vanya because of technical issues. Mm-hmm. And so what we're going to be doing now is we're going to be discussing again from a sponsorship point of view, Macmillan Research. I'm going to have a quick chat with Jacqueline Allison about some of the stuff, and then we will go on to introduce our panel. So we'll talk to you again very shortly. So yes, thank you very much. This has been absolutely fascinating to listen to some of these discussions. And there is still a lot of work that needs to be done in the upper airway because it is the weakest point of the virus. If you can control the infection in the upper airway, it doesn't get into the lungs, and therefore it can't cause damage in a significant way to the rest of the body. So we'll be discussing this with our panelists very shortly. But before I do, I'd like to have a quick chat with Jacqueline. How are you, Jackie? Your mic is muted at the moment, so I don't think anyone can hear you.
Hi, Philip. Yes, thanks for introducing me. It's a pleasure to be here.
Wonderful. So what I'll do is just so that in case people had come on a bit late, I'm going to very quickly play the sponsorship video again and just ask you a few questions about this work that needs to be done. And I'll ask you to introduce yourself as well.
Sure.
Macmillan Research is one of the Vejon Health group of companies, which comprises health and media businesses. Macmillan Research, along with Dementia Crowd Research Limited and VibroScience Limited, focus on innovative medical research to find solutions for a selection of common diseases as well as strategies to manage them. At Macmillan Research, we take a unique approach to medical research, applying the fundamentals of physiology and pathology to understand disease. Through detailed research on COVID-19, a theory of autoimmunity to angiotensin-converting enzyme 2, also known as ACE2, was formulated in April 2020. At that time, the hypothesis that COVID-19 is a viral-mediated autoimmune disease was not regarded by many as plausible. Over the past year, the theory of autoimmunity in COVID-19 has gradually gained credence. Dr. Macmillan, Executive Director of Macmillan Research, collaborated with experts to publish scientific papers in May 2020, explaining the concept of autoimmunity in COVID-19, and March 2021, proposing a more detailed and comprehensive explanation of the symptoms associated with COVID-19. The principle of autoimmunity in COVID-19 is that the viral spike protein binds to serum ACE2, to trigger an autoantibody response. This may lead to severe disease in people who have elevated levels of serum ACE2, including the elderly, and those with obesity, hypertension, heart disease, and diabetes. Sex and ancestry are also determinants of elevated serum ACE2. This theory of autoimmunity could explain the symptoms, timeline and associated comorbidities in people with severe COVID-19. A COVID-19 research project has been designed with the aim of advancing our understanding of immune dysregulation in COVID-19. This is the primary cause of lung damage which can lead to death. The outcome of the research project has the potential to change the course of the pandemic.
So yes, thank you very much, Jackie. So if you don't mind just giving a quick introduction about who you are.
Absolutely. Yeah, so Jackie Allison, my background is largely capital markets and communication. And so recently became a director of Macmillan Research, and wonderful opportunity to talk a little bit more about the research project that Dr. Macmillan just mentioned. And of course, this is so important in really understanding how COVID-19 works and its interaction, as well as that autoimmune theory, which is gaining traction. So pleased that it is doing so. But what we are really keen to do is have this COVID-19 research project moved forward. And so we know that Philip and other experts have been collaborating over some time now to discuss what's really needed here. And some of those other experts, Philip, one of them being Dr. Bruce Uhal, professor at the University of Michigan, who has co-authored papers with you. And you mentioned those two papers in that video, very important papers explaining the theory of autoimmunity and how it explains a wide range of symptoms. But there is more work to be done, as we've said, in understanding more about how this works and some of the details. And that understanding is what will help us, we think, to finally get out of the pandemic. It's about treatments as well, but it's about understanding the underlying disease. And that is what Macmillan Research is all about.
And so if there was one thing that you would be saying to people today, without going into too much detail, what kind of support are you looking at?
Yeah, so it's key to get some funding for this research project. And when we look at the type of funding that's required here, relatively low compared to many of the research projects out there. A lot of money has certainly gone into vaccines and other things. We're talking about a range of $250,000 US to move through phase 1 and phase 2 of the research. That would get us a better understanding that could, again, then maybe move on to a third phase, but critical to get that base understanding. And so for what we see as a very reasonable amount, the project has been well defined. We've identified a lab in Europe. We've identified the experts who can lead this project forward on a lab basis and get to that understanding. And so we will be providing more information about how those who are interested in supporting this research at any level, anything would be very helpful to get us to our target. And that's, again, $250,000 US.
Wonderful. Thank you very, very much, Jackie. And what we'll do is we'll move on forward now into the rest of our discussion. So we have our main panelists to come up, and it's with great honor that I get to introduce all of them here today. And so we have John Abels, and we have Christopher Newton, and we have Jennifer Smith. So if you don't mind, it would be wonderful if you guys gave a little bit more of an introduction to yourself. So I'll probably start with you, John, and ask you to tell us a little bit about yourself.
Okay, my name's John Abels, and I'm originally, as I said earlier, from Southern Africa, from Rhodesia, now Zimbabwe. My degrees are all from the University of Birmingham in medicine and in pharmacology. I had a career in practice and then in the pharmaceutical industry as a drug developer with some major pharmaceutical companies that brought me to America, after which I went out into the venture capital world and helped to develop early stage medical companies of many different sorts. But my current activities are focused on immunology-based companies and also neuroscience. And the event of COVID, has engaged my deep interest, and I have some projects at some companies ongoing in that regard, none of them in the nasal and oral delivery routes. But I'm very intrigued by what I've heard and by what I already know about that physiology and that pharmacology.
Wonderful. Thank you very much. And Jennifer, your mic is muted at the moment. If you could tell us a little bit about yourself.
Yes, thank you so much for having me, Philip. So I like to say that I'm a 3-degree microbiologist. So I have a master's degree in medical microbiology, and I earned my doctorate in microbiology and molecular cell sciences with an emphasis on virology. So my career has been spent mainly working with influenza virus. However, I've worked with other respiratory viruses, paramyxoviruses, RSV, and I did some work on an Ebola contract looking at testing vaccine candidates there. So a lot of my work has been in vaccine development and testing. When I was at the University of Georgia, I spent most of my time working on aerosol delivery of influenza virus into the nasal passages as a mechanism for vaccination. So this is very interesting to me, looking at nasal delivery systems and bioaerosols.
Wonderful. And yourself, Chris?
I started off in the cancer field. I did my PhD at St Mary's in London, and I was working on endometrial cancer. Then I moved to the Marsden, working on breast cancer. So I was developing anti-estrogens, aromatase inhibitors, and this sort of thing. Then in the 90s, as a family, we moved to Germany for 6 years, and I spent 6 years. It was a pituitary group, so neuroendocrinology group of the Max Planck, and unfortunately, I think to the dismay of the director, I carried on working on sort of breast cancer related stuff and looked at the estrogen receptor and worked on the activation of the estrogen receptor and how it could actually be functional without estrogen, so it could be activated by growth factor pathways. We came back to the UK. I worked at the university in my hometown. Moved then into, I set a small company up, worked 10 years in that, and then really I've been sort of hijacked by the Lyme disease group, I suppose, and trying to help them out. And there's so many parallels between Lyme, for example, chronic Lyme and chronic COVID, if you like, or long COVID. So I was working with a local clinic, and I wanted to talk with Debbie, especially because we developed a system for, it was an electromagnetic system. So I really want to question her on what she said earlier.
Yes.
And I really got into this because I was waiting for a hip operation, which was being cancelled. And so I thought, well, if I can do something about this and sort of put an end to this quicker, I'll get my hip fixed quicker. But of course, we know what's happened. I've got my hip fixed, but whatever we say doesn't seem to make much difference, unfortunately.
Yeah, wonderful, great. So before we talk with any of the presenters, I will probably start with Marcus first. Generally, because what are the thoughts with the upper airway now? Because some people are coming from a background of not necessarily knowing a lot about it. So before we come to Jennifer, which your mic is muted again, let's start off with John and Chris. Your thoughts on upper airway in COVID-19.
Well, I think for my own part, I think that if the relevance is going to be there, is in terms of practicalities, it has to really focus on prophylaxis and very, very early disease, almost before any symptoms develop. Because by the time symptoms develop, I think that the virus already passed into the body, causing the, temperature rise and body aches and the general flu-like symptoms of the disease. So that if you're going to try and interrupt the viral propagation at a disease stage, you really have to come in almost at day one, if not day zero. So from a practical point of view, that may be a problem. From a prophylaxis point of view, on the other hand, it's more promising. Now, there are already things on the market that have been, both of them, developed in Israel, and one by a company called Sanotize, which is really a nitrogen NO gas, device and treatment, and the other called Tafix, which is a form of gel. It's actually in a powder, and when it reaches the mucous membrane with the aqueous and mucin, etc., forms a gel barrier to the nasal mucosa, and can act as an extra blanket, as was mentioned in the presentations. So these products are already out there. I don't know if they're already out in every country that I know, but Tafix is available in the United States, and it's being marketed. So there already are approaches, and there are so many other molecules and interventions that could do the same sort of job. There are people in the holistic medical environment here that are using dilute hydrogen peroxide nebulizers as a treatment and prophylaxis. There are other things that are non-gaseous or non-liquid, things like copper probes. In fact, I bought one. In the very early days of the pandemic, I thought, well, gee, there's these copper probes you can get, rounded probes you put up in the nose and twirl it around for a while, twice a day. And copper ions are then released in very, very tiny quantities, copper ions being extraordinarily potent antivirals against COVID. And so there are already available products out there. And I think their real meaning will be in the prophylaxis side rather than the treatment side. And then all the way across, the approach to the nose and nasopharynx with vaccinations, I think, are very promising.
Yeah, that's
the
one.
natural ingress of the virus is through the mucosa. The mucosa has its own assembly of lymphocytic and other immune responses and dendritic cells and all that sort of thing. And by educating the immune system at that level, one then can get a sort of a gradualization of immune education, which I think is more natural and probably could prove to be more acceptable, fewer adverse effects compared to the injections of codons that we're using now, and more natural induction of a natural immunity, which I think is much more broad and much more effective against variants.
Yeah, this is something I'm going to specifically be asking Jennifer about, but I'll just get Chris's thoughts generally before I ask Jennifer about nasal vaccines and so on.
Yeah, I think really what we're talking about is sort of DIY, aren't we? We're not looking at sort of broad uptake of these things, but it would be a sort of situation that, if you go out to the supermarket, the local shops or something like this, you spray some of this stuff. Or frontline workers, maybe in hospitals, would perhaps use it. One of the questions I wanted, if I can I ask Stan a question now?
Oh, don't worry. Yes, not yet. We'll bring Stan in a minute.
Okay,
so hold that question. So what we'll do is we'll capture some thoughts from Jennifer about generally nasal vaccination, because that was the bit that I'd hoped that Vanya was going to focus on. And so, Jennifer, any thoughts with this area that does seem to be underfunded and less attention has been given to it?
Right. So that was some of the work that I was doing at the University of Georgia. So we know that for influenza, there's an intranasal vaccine. MedImmune had one called, it was a live attenuated influenza virus that was delivered via syringe into the nasal passages. So I think that was taken off the market. There were issues with this. So when we think about delivery to the nasal passages, and I think Marcus touched on this a little bit, is that delivery systems are very important. So the delivery of the intranasal vaccine impinged on the vaccinator. So depending on each person who was giving the vaccine, it was being delivered at a different rate, different droplet sizes, issues around the droplets either being too large and dripping back out of the nose or getting back into the throat and being swallowed. So the dose that each person that may have been getting would be variable, right? So each person, so there was a lot of complaining about this vaccine not being very effective. Now we know that live attenuated vaccines are very effective. It's the delivery method that was the issue. So what we were doing was using a small particle nebulizer to deliver the live attenuated virus into the nasal passages. So the issue would be so particle size is very important, right? So we know that certain particle sizes, 20 microns, will deposit in certain areas. Smaller particle sizes, less than 5 microns, are respirable, so those will get down into the lung milieu. So we want to look at particle size and then also deposition. So where would these particles being deposited in the nasal passages? So the value of that is then what type of response will you get? So I know we want to talk about mucosal immunity, right? So getting the virus or vaccine or treatment delivered up into the nasal turbinates or past the nasal turbinates would be very important in terms of getting an effective mucosal immune response. Right. So we don't want to just get it right into the inside of the nares. Right. That is key to getting an effective protective response and also for therapeutic. So those are some things that we need to think about. Something that I thought was
also, Jennifer, isn't there also, and I know this applies to other intranasal drug deliveries, like the attempts at insulin through the nose, etc., that the health of the patient at the time is also very important, because if they're suffering from a cold or something, they're producing so much mucin that you can hardly get anything past that.
So that's actually what I was just going to mention. So for SARS-CoV-2 infection, we know that those who smokers are more prevalent in suffering from severe disease, right? So what happens there, as Marcus mentioned, is we have mechanical barriers in the nasal passages, right? So there's the mucociliary in the nose, and those are very important for trapping particles. Things like dust and smoke and stuff like that. So when people smoke, the cilia get burned off, and so that one mechanical barrier is now taken away. So they're more susceptible to infection. So so that goes to your point about different immune status and also health of the person in these therapies.
So you mentioned, so I just brought Marcus in. So Marcus had done the presentation with regards to the sprays and the technology with regards to that. Any questions for Marcus?
Okay, so Marcus, have you looked, you mentioned chlorpheniramine. I have some concerns. That's a very active substance. It's an antihistamine. It's a drying agent. It's anticholinergic. It's all that sort of thing. So using that, would, to me, be more complicated than using very simple sterilants. It's funny that we're talking about this, because in a way we're validating what Trump said, about using sterilants, to treat COVID. And I think he was misquoted and probably had a point. But so sterilants like hydrogen peroxide dilute or hypochlorite dilute, or as was mentioned, povidone iodine dilute, wouldn't they be more easy and effective also in your systems, rather than chlorpheniramine or using hydroxychloroquine or anything like that?
That's a great question. Actually, I have basically two parts to it. In the formulation, that's the first thing we think about particle size. We think about concentrations. These are concentrations on confedramine that are effective, yet they are not toxic, even from all the toxicological experiments. But there's a very important detail here. The distribution of the particles is key. We want to spread those molecules evenly. So many of these sprays that we have in the market, basically, they don't take that into account, and they accumulate in certain areas while not providing the protection to others. And the second part is we have to remember that this is just the beginning of the disease, let's say in the nose, and we can stop it there. However, past beyond the 7th or 8th day that we can recognize that this turns into from the viremia, we go into an immune dysregulation, allergy-like syndrome. So the properties of confedramine allowed us to have that anti-allergic, anti-inflammatory potency. All we need to do is decrease the particle size and distribute in inhalation. All the experiments, all the studies, again, looking into confedramine and the benefit it has while maintaining those very low doses. And it's interestingly, some of the cholinergic effects are in very high concentrations. So today, for instance, if we have 1.5 milliliters, even if you open this and drink it, there's not going to be any toxic effect beyond a little bit of drowsiness and a good night's sleep. But all of those aspects have been taught, and there's value for the hygiene and disinfection strategies. But again, compared to medications, they can be applied at different stages of the disease. Now, if I take the confedramine, and in this case, I can use xylitol. Xylitol have all these other properties that are friendly for the microbiome, and at the same time, it's tough against the pathogens such as COVID. And that will take us into a very different, let's say, more preventive approach. So we need to have the strategies in terms of where, at the point of what we are attacking the disease, preventing, intervention in the nose, intervention in the lungs. We're attacking the virus, or we're attacking the inflammation syndromes, immune dysregulation caused by that. So the nose and the inhalation therapy and the gases per se offer all those benefits compared to systemic, let's say, treatments.
What you said about inhalation in the lung is very interesting. In the later stage of disease, and Philip and I have discussed this ourselves, there are now quite a lot of interest. There's a lot of interest in using antihistamines like chlorpheniramine, but other ones have been mentioned. Promethazine is the one that's been used mostly by Dr. Chetty in South Africa, who has promulgated the idea that the dysregulation after the initial viral phase of the cytokine storm hyperinflammatory condition that patients enter. And Philip has pointed out to autoimmune mechanisms, which I think both that mechanism and also what Chetty invokes, which is a type 3 hypersensitivity reaction. And hypersensitivity reactions are, after all, autoimmune. The use of inhaled antihistamines is, this is the first time I'm hearing this idea, is pretty intriguing, and if you mix them together with steroids as inhalants, then you really have something. It could be very interesting. But, we're talking here about nasopharyngeal early stage rather than that latest approach. And in that regard, I would say that chlorpheniramine could be used. Other drugs could be used, but not hypertonic saline, hydrogen peroxide, hypochlorite. Even referring back to the presentation on plasma inhalation, which is invokes oxygen singlets as a sterilizing agent, as do many of these things, like povidone iodine, invokes iodine singlets and that sort of thing. So we need to focus back down on the early stage and prophylaxis, it seems to me.
Yeah, I just wanted Chris to get in his question because I wanted to bring Debbie in quite soon.
Yeah, it's sort of two parts. I mean, I think one of the fundamental problems, as we talked before, is we know so little, and the reason about the early stages of COVID, the so-called symptomatic stage, which is a ridiculous title because it's the whole thing symptomatic. But there we are, the first sort of 7 days first week post symptoms started, and that I think is such a fundamental problem, and it's because the medical system has gone AWOL. Basically, it certainly has in UK. It's basically told people to go away, so they haven't got information as to what's going on, and certainly the detailed information in terms of changes in immune cell fractions or proportions, cytokine, chemokine changes, and I think this would be so useful with respect to these early treatments to lower. I mean, we're talking about lowering viral load. I guess we're not talking about destroying the virus, and so that's sort of part of my question. Wouldn't it not be? And I'm sure you're looking at this, but wouldn't it not be sort of sensible to include things like vitamin D in these nasal sprays? So you take in a compound that is actually going to activate the cells that are going to do the cleaning up. So the macrophages, the neutrophils, this sort of thing. So you go with a multi, because I always think this thing can't be sold with single agents. It has to be a number of agents together. So if you could get appropriate vitamin D analogues into the sprays as well. I mean, one of my good friends who sadly died recently was using a sublingual spray for vitamin D. But I mean that could be included in your Marcos and Stan's products. The question, sorry, reducing viral loads. PCR, such an awful technique, really, to look for viral loads. How do you actually measure viral load with PCR when in terms of active virus? Because if whatever you've got in your formulation damages the virus, you'll still measure the RNA unless that short segment is destroyed, which it could be. But you need to look for a number of different segments. After all, with PCR, we're only looking at sort of 100 base pairs. It's not like normal PCR. I don't know how people in papers justify measuring viral load by using PCR.
Any thoughts, Marcus?
No, I'm going to give that to you because it's basically what's being taken to understand these potential mechanisms. Again, that's one marker that we look at. We also look at the clinical progress of the patient. It's in the context. Like I like when people tell me, I went to the lab to draw some blood. What's the indication? We need to know in the context of. In terms of other signs and symptoms of the disease, that using these therapies and drugs with these special properties like anti-inflammatory, we have seen patients that in the anosmia phase that seems to last between 7 and 12 days, we can reduce that by half. So suggesting an anti-inflammatory mechanism that is occurring at the level of the nose and the nasopharynx. Now, in terms of viral loads, again, that's our best shot right now. We are looking into a sequential quantitative RNA. Might not be the best tool that we have, but in terms of the way we conduct these, we are thinking FDA mentality, in the sense that here's an indication and here's how we're going to treat it. We need to have the mechanism tied to that particular indication. So the way we think this is working, and we propose, and we're testing this, is that it decreases those viral loads or those virus particles in the nose and the nasopharynx. So that's the best tool we have right now. And before that, mixing and the different formulation, it's a fascinating idea on basically add some substances like vitamin D that they have properties that can help decrease the inflammation or add into the host mechanisms to eradicate the virus. Again, I'm not going to this area. I'm not an expert in microbiology because right now we're getting into that sense of how we can support the microbiome the same way you give vitamin D to the host. You can give probiotics and inject, or we can distribute in the nasopharynx probiotics. One example, very quick, one example of those are a lactobacillus. You provide enough a salotube lactobacillus, it cannot digest it, but it's allowed lactobacillus to produce basically fungicidal properties, because lactobacillus doesn't want fungi there, and then you have rhinitis owing to fungi. And so that portion in terms of experimentation and development, in which we now take the microbiome and make it work for us, it remains to be seen. We need more research, particularly in that area.
Wonderful. I'm going to, oh, you're going to ask him a question. I'll tell you what, Jennifer, can you hold that question? Because we're going to bring them all together, but I want to bring Debbie on so we can ask her some questions, and then, or do you want to ask that question first? Go ahead. All right, go ahead.
Well,
Jennifer.
I just wanted to say that I'm glad you brought up the anosmia, because that's what I was thinking about. One thing that I found interesting doing case investigation was the number of people who experienced the loss of smell, loss of taste. And early on, I thought it was a rare sort of event. However, as time went on, it seems at least 40 to 50% of all people experience this anosmia and ageusia. So I find that very intriguing. And so I'm glad you brought that up in terms of treatment, because, I have a lot of people who complain that they lost of smell, loss of taste, so they lose a lot of weight because they don't want to eat. And getting that resolved can take a long time. So I like that you brought up that as far as how your product may affect the bringing, resolution of that sort of thing. The other thing was congestion. So I don't know in terms of treatment, if you looked at, because last year I actually think I had the COVID early on. And one of the things that was really difficult for me was the congestion, because I literally couldn't hear anything. I was so congested. And so how would your product have an impact on that in terms of treatment?
It's very interesting. I mentioned at the no silver bullet in medicine, and there is not. However, confentermine is one of those things that they have various properties. We mentioned anticholinergic, anti-allergic, antiviral, and there's a particular pathway that it inhibits the aquaporins. So it has some drying effect as well. It's interesting, we're doing research in Dominican Republic, and the latest wave is characterized by a very aggressive nasal congestion. This, I'm not aware what specific variant they have down there, but I'm very aware that clinically expresses that way. And here is the other portion of it, that using the intranasal route, and as we've been working with other scientists, Dr. Basu from UNC, not only the molecules, where we put them, how we put them, in what amounts, how they diffuse, it's part of the whole formula, and it takes, again, in the development portion, a lot of interest. Now, coincidentally, we can identify other particles or molecules that they can offer different benefits depending on the indication. So COVID has the whole 9 yards in terms of different manifestations. We have a very case in Venezuela because of the variants there. It's long-term anosmia. So I'm talking over 30 days, and as you mentioned. So maybe the anti-inflammatory actions, maybe less damage to the nerve. Again, we don't know, because we are so focused on the virus right now that these other things we are pretty much overlooking.
Yeah,
I mean, we know that anosmia also happens in other respiratory diseases as well. So
I agree. I'm going to pause this here so that we can discuss some of the other ideas. And remember, Marcus will still be here, so we can come back to him. So I'm going to put Debbie in the hot seat at the moment. I think, Chris, you probably wanted to start off because you're quite interested in the technology. What have you got to ask?
Yeah, absolutely. I mean, I should just say that when I was sort of commandeered by the Lyme disease fraternity, I was working with a local clinic, or a lady who retired recently as a GP, with people coming to her with what they thought was Lyme disease, and we actually set up a device. It was basically a sort of Rife technology. And this is why I wanted to ask you, Debbie. I was not exactly clear. So the system you're using, is it a combination of a sort of so not oxidant generation, but there was the frequency aspect here, because I don't, the system we're using, I don't know how it works, and I'm not sure anyone really does. But it was set up originally to use radio frequency. And then over the years, people started to use audio frequency, but to provide it as a magnetic pulse. So does your system combine the two? Does it have so you're generating somehow fluxes or variations in oxidants? And is there some kind of, you were talking about the interaction with the body, and also the fact that you were using a resonant frequency, which is what supposedly this system that we built and others have used is doing. I've never been able to do the experiments to actually show that, but others suggest that it does cause a resonance. So is your system destroying, I was going to say, have you been able to do it? I'm sure you haven't, but I'm sure you'd love to be able to do it with a virology setup, to see these viruses actually being physically destroyed at certain frequencies. So is there a frequency element combined with the oxidant administration?
Yeah, thank you, Chris. Yeah, I can answer your question. We've actually been doing this for a very long time. So for 53 years, this device has been on the marketplace, and getting rid of pathogens, bacterial and viral and colds and flu is the main thing that we do. It's the main reason people purchase the device, and it's more expensive. It's not a cheap option for the consumer, but it's an option that they'll have for many years. It produces, it operates at atmospheric pressure, non-equilibrium atmospheric pressure, and also atmospheric temperature. And it produces reactive oxygen and reactive nitrogen species, and also oxygen singlet species, which, as you may be aware, operates very differently once it's inside the portals. And we emit that off the needles. So the machine's got high voltage inside. Off the needles, it comes out in a vortex shape, and it flies through the portals in a specific way to reach the upper respiratory system. So because our patients use our device every night when they go to bed, it is actually clearing their upper respiratory system on a nightly basis. And so this is why our patients say that they just don't get colds and flu anymore. It's the most common comment we hear. And yeah, it really is quite strong in how it does that. It doesn't matter what mutation it is either. All mutations, we seem to get the same result. It doesn't matter what age the patient is, from a newborn to an elderly person. It doesn't matter what comorbidities they've got. It works on all of those without hesitation. I can't say that it works on all mutations of COVID-19 because I've only been exposed to treating patients in Wuhan, January, February 2020. And here in Australia, we've had very little COVID and very little mutations yet to start treating.
So this is a very physical process. It's oxidant damage, in other words. I mean, it's rather similar to the ozone story in many ways. Yes,
it's similar. Yeah, I would. That's what I want to ask. I mean, if you're advocating, I mean, as you say, some people with upper respiratory, chronic conditions, asthmatics, people with chronic bronchitis, et cetera, et cetera.
Yep.
I mean, if you're advocating the use of this nightly, and sometimes during the day, what kind of side effects are you getting from continuous oxidant exposure?
This is a good question, John. There is no side effects because of the way we do it. Our device is very, very gentle. Like I said, we've treated over 1 million people in 53 years. There's been absolutely no recalls. There's been no adverse health events from our patients. And we're treating a whole host of different illnesses as their health changes throughout their life. They call us and they say, what frequencies do we use now? And what do we do now to control this or that? And, you were talking about Lyme disease and, I only this afternoon, well, it's now 3:30 in the morning on a Sunday here, but it was a few hours ago now. But yeah, just recently had one of my most severe Lyme disease patients contact us. Very excited she was, and it's taken 3 months now for us to work on some really severe problems she's got on her skin. And she sent me some photos, and she's doing really great. So yeah, we've changed it. It's really, it has to be assessed on a per patient basis. So there's 144 medical settings in just this little one here, all different frequencies. Our frequencies are very low. They're very safe for the human body. So they start from, very low 1 hertz up to 30 hertz. So we're not even talking anywhere near the frequencies of other types of devices in the market. We don't really have any competitors at all at this point in time. We've sort of had a market of our own for a long, long time, but our inventor just wanted to keep it in Australia and really didn't because we'd never published our results. In fact, tonight is the first time in 53 years we've actually released some of those results. Yeah, it's.
I need to understand this frequency notion. It befuddles me. The frequencies you're referring to are the pulsed EMF that you're using within the machine to generate the plasma, correct?
There's a number of different
what are the alterations in frequency really influencing? Are they influencing the plasma itself? Or are you hypothesizing that you're essentially extending electromagnetic fields to the patient nearby from the machine?
Okay, maybe if I explain a little bit more, because our science is very, very complex. To begin with, the high voltage behind the needle generates the plasma on the needle. And then the plasma has a plasma plume. And it's at nighttime, you can actually see it on the needles. And that plasma plume is a beautiful little glow. And inside there is the beginning of the production of the reactive oxygen, reactive nitrogen species, and other types of species. Then it travels through the portal. During the portal is pulsed electromagnetic fields, but it actually goes through an electrical circuit. And inside the portal, the species are controlled. So, for example, the negative charges of the oxygen and the nitrogen, they will go through the portal as per normal in accordance with the frequencies selected.
Can I just ask what the portal is? What is that?
The portal we generate special portals for the human body. So we have one portal that takes our species and our pulsed electromagnetic field from the device into the human being. When those molecules and those ions reach the human being, they have a very unusual behavior. And what happens is inside the human body, the molecules and the ions are vibrating and shaking, and they're spinning really, really, really fast, especially the oxygen singlet, which is actually at the edges of the field because it doesn't get attracted to the charges. It's neutral. So some of the species are neutral, so they won't go through the field. They'll go to the edges of the field to reach the human body. Once inside the human body, the molecules and the ions start doing their thing and getting rid of, eliminating pathogens and doing all the different things that they do within the human body. But at the same time, we've actually caused, that's a pushing effect. And then what we've done is we actually cause a pulling effect. So we're actually forcing the electron to be ripped away really fast from that molecule inside the body. So it's only the negatively charged oxygen ion that's actually able to transfer its energy. But then coming back from the human body, back into the circuit, it's a magnetic pull of that electron to pull it back out of the body. Just like you go and stand in the sun and you'll have 10,000 muons go through your body every single second from standing in the sun. We cause the same effect to happen, but with the electron. So we control the flow of the electron and where it goes and how it comes out of the body. Why are we doing that? Because when the electron and the molecule are together and the ion inside the human body and they're starting to do their reactions, when you rip one away, it causes a massive, at that very point in time, it causes a massive, what do you call, it's like a restriction, like a, they I've forgotten the word, but they compartmentalize. And so when this happens, it generates protons and light. And those protons and lights actually make it's a collision, sorry, that's what I was thinking of the word. So when that collision happens, the protons are released, the light is released, and that further enhances the antioxidant nature of the human body. It's very, very, very complex, and I've actually asked some of our scientists if they can put all this into a paper for us. It'll probably be our first paper written.
So is it?
But in the context of COVID, I think it's all, if you really want to get ahead on this, and I'm talking about business now, as Philip thinks, I'm really not a businessman. I'm more of an investor, and I continue to be try to educate myself deeply in science. But I think in terms of a regulatory approach, if you really want to get an official approval, and as I say, you don't have an official approval for COVID or other respiratory, upper respiratory diseases, all you do is have to invoke a sterilant effect, against the bacterium. Once you get into this, vibrating molecules and molecular memory and water, which I know is a phenomenon, but once you get into frequencies and ion smashings and all of that sort of thing, you're getting into very new agey kind of explanations. They may be very, very interesting to theoretical physicists, but in terms of product development in the medical arena, I think you stick to the fact that you're generating plasma which has a disruptive effect on air molecules and causes reactive species, of oxygen and nitrogen that can be sterilizing gases or sterilizing within the nasopharyngeal and upper respiratory tract. Once you have that, you can really start to then have a rationale that the feds would understand, and then do the necessary clinical trials, with the dummy machines, at night, double blind, and then to see the differences between those two groups. And then if you establish that the treated group have a significant difference, from the untreated group, then you've got yourself a real product that maybe doctors will prescribe.
And you've just had a consultation there with a business consultation. That's quite fascinating, John.
No charge.
We do already have that on our TGA certificate for colds and flu. So we are already registered
in the United States, or where is that approved?
In Australia. In Australia, we are already registered to treat and prevent colds and flu of the human body and blood system. And the
same thing would apply to COVID. You've got to go through the studies and get those ready.
Yeah, that's
and
right.
Then if you get it in the I hate to say it, but the FDA is still regarded as the sort of the primary regulatory body, and if you get it done here in the United States on the clinical trial basis, then you will have achieved a lot.
Yeah. I'm going to have to pause here so that we give some time to Stan at the moment. Any questions to Stan?
Well, Stan, you're close to me in geography. I have a home in Boca Raton, just up north from you. I'm not there now. I'm at my place in Connecticut. But when I get down in the fall, you and I should meet. Povidone iodine you've mentioned as a molecule. I know of other iodine, not only iodine but also chlorine species would fulfill, like swimming pool bleach. Hypochlorite, which generates chloride.
Careful, Trump said that.
I know. I don't want to be political, and I don't think any molecule should be political, whether it's hypochlorite or hydroperoxide or hydroxychloroquine. They're independent of politics and should never have been drawn into a political gambit here. There was a great pity. But seriously, the approach of misting nebulizing a sterilant of that kind, whether it's a bleach sterilant or non, is very attractive. The question is, what is the remaining market, Stan? How many patients will there be who have not had natural herd immunity or have been induced immunity by monovalent or in fact polyvalent vaccines? And to what extent do we really need these now, as these other interventions have sort of created natural resistance in the first place?
Yeah, I think that's a really great question, John, and would look forward to getting together. The way I look at it is that there are going to be some populations that will not get the vaccine, and there may be some point in time in the near future where other variants emerge that may be resistant to the vaccination. And working for GSK as a company, a big vaccines company, I realize it takes a lot of time to come up with various other boosters and so on. So my general view is, this is still, unfortunately, an area that people have not recognized. Unfortunately, I have to say that sales are not that great at the moment, as like other things with the nasal cleansing, nasal sterilization. But I do think there's going to be, a small portion of people that will use this, that will be vigilant, just like people that currently irrigate and other things in nasal cavity. And so I think that will be the market, the people that are really hyper vigilant around cold, flu, corona, whatever it may be. To your point on hypochlorous acid and other things, the great thing about PVP iodine is its stability. Hypochlorous acid and hypochlorites are very unstable, so they have a very short shelf life. Interestingly enough, when you look at the hypertonic plus hypertonic salients plus surfactants and other things that have been done, they're shortening cough by like 5 days, 10 versus 5. They're shortening nasal congestion suffering from 15 to about 7. And there's a thought here that it's not only about just rinsing and cleaning and reducing the viral load, but hypertonic saline is believed to create at the kind of tissue level low levels of hypochlorous acid.
Absolutely,
right, which we know is
and chlorine ions, basically.
Right, chlorine ions. So these things are probably benefiting, the kind of sterilant effect. And back to what Marco had said about, general levels of nitric oxide, which we know is actually quite good, usually in the nasal cavity and obviously a little bit in the lungs as well. So I think I do believe I'm a very simple guy in that, I realized that when I was working on this, I wanted something that could be implemented immediately because people are dying every day. And so I looked at, to your point, what's the simple science? What's the best approach possibly you can get there? How do you address the consumer barriers? But also to your point about regulatory barriers. How do you get this thing to market without having to educate the world? Just educating people on why they need to clean their nose is a big task. It took how many years for physicians to learn to wash their hands before seeing other patients, right? To reduce hospital infection. So, what we're dealing with here is, unfortunately, sounds very simple, but in practice and in consumer and people's behavior is not that simple.
Mm-hmm. So I have a couple questions, and I don't think you addressed this in your presentation. One is the delivery mechanism. How are you delivering this in terms of, okay, so in a simple...
Yeah, so it's a mister, nasal spray misting,
okay,
right? And then a targeted delivery for the throat and mouth. So you pop off the cap and then you insert and spray to the back of the throat, swish it around and spit it out.
Okay.
I mean, iodine is very safe, but you don't want to eat a lot of it if you can help it.
Yeah.
So then there is the
septic effect. I don't know if you saw recently, there was a group of parents that sent their children's masks to a laboratory to have them tested, and they found all type of pathogenic bacteria. They found TB. They found a lot inside of these masks that these children have been wearing, and that's that's troubling. I think it goes to your point of, cleaning the nasal passages like we do with mouthwash for oral sanitation, and brushing our teeth. So I think that is very important in terms of getting rid of some of those pathogenic bacteria and microbes. So I'm curious also about the mechanism of action. I'm not really familiar with PVP iodine, so I don't know if you could.
Yeah.
I don't know if it's a simple.
John touched on it before. It's a broad spectrum antiviral, antibacterial, because it contributes free radical iodine.
Okay.
The iodine is what kills it. Right. And the data I have on COVID-19 U.S. Strain is it kills with our solutions 1% PVP iodine, even diluted out, kills COVID in 5 seconds to about 4 to 5 log. So you almost can't measure it. It's so fast. Right. So we purposely made it at 1% because we knew intuitively that when we spray it in the nose, it'll be diluted out by the mucus, and there's also bioburdens and other things going on. But it hangs up in the nose for about 15 minutes.
Stan, how have you actually measured the killing? When you say killing, what do you actually mean by that?
What I can do, Chris, is I can send you the paper. It's just standard. It's a standard viral where they use embryos to check eggs and so on.
I'd like to bring up a provocative question. We're ignoring the eyes in all of this. The eyes have been established as a portal of entry for the virus too. I don't think it's necessarily more of a portal of entry than the nasopharynx. It may be very, very subsidiary in that regard, but still, when people are, when they adopt N95 masks, they goggle as well because of that portal of entry. So, what are we missing something by ignoring the optic sterilization?
Yeah, it's a really good point. And in fact, the one study that was done by Chowdhury in Bangladesh used a nasal spray, an oral spray, and an eye drop in that. And I think that's right. My approach did not include an eye drop, although I'd love to do it because it's currently RX. It's used in ophthalmology for quite a lot, of years for surgery. And the only reason I didn't bring that to market was I couldn't do it in a rapid time frame because it's an RX product in the U.S. And around the world, it's pretty much a medicine. Also, when you look at the prevalence of infection, based on everything I've read and talked with other experts, so to speak, the nose is probably the biggest point of entry, probably the biggest one. And so we kind of went at it, 80-20 rule. Yeah. But I would have loved to have done an eye drop.
I think it's a good question in terms of the contribution of the different routes of transmission and infection, but I do think that as far as the ocular route, it's probably more people touching their eyes, rubbing their eyes or touching their eyes more than just droplets landing there.
Or aerosols.
Yeah, I'll tell you what I want to do now is that we've just about reached the end of our defined time frame, and I suspect there may be some more questions. I haven't touched any of the many questions that have been put in front of me, but what I wanted to do was to formally close our meeting, and we can just keep on talking for as long as there are any questions that people want to deal with. Is that okay?
I'm
So I just wanted to say thank you very much to all of our presenters and our panelists. I think this was actually fascinating. I spent most of my time just listening and playing with the controls. I've learned so much hearing from everybody, different angles, the different ways that we can treat. And I think this was the important thing that I wanted to get put out, is that there are many tools in the toolbox. We get success when we start to utilize as many as we can. And managing COVID, as I think it was Professor Banerjee in India had said, this is not a sprint, it's a marathon. And so therefore we have to recognize that all the techniques that we are developing will be relevant at some point in the future, because it's likely that we could be battling with this for some time. So what I want you guys to do for me is, well, I guess thank you. I'm formally saying thank you. Just stay where you are. I'm just going to do the closeout outro video, which will be about 1.5 minutes, and then we will continue to talk. And whoever wants to remain with us, that would be wonderful. Thank you again to Macmillan Research as our sponsor. And again, thank you very much to everyone. Just hang in fire for me. Yes, and thank you again, guys. So I brought everybody in, and if there are any general questions or things you guys want to talk about, I guess I'll ask the first question. It will be interesting. Jennifer, do you have to run?
No, I have a question.
Oh yeah, sorry. Go ahead. Yes. Yeah.
So I just want to clarify or see if I understand, Debbie, your device. So is there a liquid or something inside of the machine?
No.
And then the needles, are the needles like different types of metal, or is there a specific metal, or what are they made out of? That I don't know. I guess I'm trying to understand how they're generating this.
Yeah, it's as I said earlier, it's actually a very unique device in the world. I know it's still the only one in the world of its kind. The professors have told me that. The needles are specially made. They're made of a mix of different metals. They're also got pure gold in them, like most advanced electronics do. And they sit into pure gold beds as well.
Okay, so there's no liquid or...
No liquid, no filter, no motor. It's just high voltage power and a lot of very specialized engineering.
So it's more like sort of an air purifier.
I wish it was just an air purifier. It'd be a lot easier to make and a lot cheaper. But no, it does purify the air, but that's the side effect of what it does.
Okay.
Think of it more like an air purifier designed for your body and your blood, because the patient has to be within 1 meter to 2 meters of the device. The species won't live past 2 meters. And so you need to be inside the biological range for it to affect your body and blood.
So vibrating water molecules in the air close to the person.
It actually bombards water molecules in the air. So a normal air cleaner wouldn't. Normal air cleaners would attach to that, and they get heavier and heavier as they move past the source, and then they fall to the ground. In our case, the molecules are so small and so tiny. We're talking about 5.64 times 10 to the negative 17 in size. So it's really, really, really small, like what is that, 44 million times smaller than COVID-19.
So what's happening is a very high field of electrostatic discharge that generates charged electrons, negatively charged electrons. And those negative charged electrons immediately react with anything nearby and then impart charges to them, so they generate reactive species out of the air. And if you can get the electrons directly to the target, they will have a direct effect again. And what happens in aurora in the North Pole is these electrons come in through the atmosphere and then interact with air and water particles. And as was mentioned, there is a there's a photo effect, with the luminescent effect with the electron discharge around the air and water molecule. So that it's a plasma generator, and plasma generators are known, and it's an intriguing way, in my view, of getting reactive species that can be inhaled, or maybe, I don't know yet whether they can be taken in through the skin, but that was claimed. I don't know how to prove that, but certainly insofar as what we're talking about in terms of nasopharyngeal sterilization, it would be an effective medium.
Yeah, I have already proven the transdermal effects through cancer tumors. When I was in Wuhan, as you know, it's a respiratory device, but there was patients there that I treated who were intubated, so I could not remove their mask when I was treating them. So with the doctor's permission, what I did was roll them into the fetal position, and I treated their lungs through their back. And I got success. So every single of the three, I only treated 3 patients. Every single one of my 3 intubated patients were able to have the intubation removed from their throat and come out of their unconscious state within 3 to 5 days and discharged within 2 weeks.
Can I just ask? I mean, this sounds more like it's you're doing something to make the body react rather than
immune stimulation.
Yeah.
Yeah, we do. When we cross the blood-brain barrier, we know we're able to influence the central nervous system and the hormonal system. We know that we're decreasing blood serotonin as the patient sleeps and increasing melatonin. We know we're affecting the macrophages. We know we're affecting the aquaporins of membranes around the body. We know that we're eradicating and eliminating colds and flu viruses from the upper respiratory system. We know we can also do that into the lungs, even when they've been infected, because that's how I did in Wuhan. They were serious cases that I treated. And as I said, you might have seen the film with the breast cancer and the diabetic foot wounds. You know, they're hard evidence. There's before and after with those.
So it seems like increasing blood flow and the lymph system, is very important for reducing inflammation and, oxygenation. So, that might be a mechanism.
Yeah, there are so many mechanisms.
Immune cells, part of the killing is through superoxide generation. So, so it's not difficult to perhaps generally surmise that by adding oxygen radicals to the environment, that these would, encourage further superoxide activity from the from killer cells and immune cells in general. So superoxide dismutase exists in physiological systems to negate to put a control on that kind of effect. So superoxides are very important, and other reactive oxygen species like OH ions also are killer species against invasive tumor cells or pathogenic agents, pathogenic viruses and bacteria.
I want to ask a general question, because I've noticed that everybody seems to have a perspective on this, which is, where do you all think we will be in 1 year's time regarding COVID-19? Anybody want to go first? You want to go first, John. All right, you go first.
I see several things. There are those things that will happen because of the ordinary orthodox allopathic medicine, and other things that will be happening from holistic, more naturopathic and trends. So On the allopathic side, orthodox medicine, I believe that we are going to see variants arise that are more resistant to these monovalent vaccines that are being used now, that are spike protein only, coding for spike protein, and then evasive mechanisms will happen, and so you'll have to have new kinds of spike protein vaccines introduced to cope with those variants. Eventually, we'll get to polyvalent vaccines, which I think will be much better at stopping variant propagation, and because there will be other elements of the virus construct, including the nuclear capsid and other membrane proteins in that vaccine. So that, I think, eventually will have a major effect on the epidemiology. In the meantime, there are companies now that are developing oral and antiviral agents, like Merck is developing molnupiravir or something. I've got the name wrong, but something like that, which they're showing early effects on antiviral load, and that will be developed as a drug, and then if approved, will be an expensive oral treatment, somewhat similar to Tamiflu was for influenza, and other agents now also for influenza. So that will happen as we go along. Meantime, I've been a proselyte of using extant, easily available, cheap oral medications that have come under fire again politically, and that was such a damn pity. Because these molecules exist outside of politics, right? That were researched only in hospitalized patients where we know that the antiviral phase has already elapsed, and they're now in a hyper-inflammatory immune dysregulation phase. But if we use these molecules, like the ivermectins of the world, like the hydroxychloroquines, but not alone, they are essentially zinc ionophores, and they aid in the transport of zinc into cells, which otherwise doesn't get in very easily as it's positively charged. And zinc is a proven antiviral, as many divalent cations are, including copper. And so by transporting zinc into the cells, they do affect proteases and replicases and have anti-inflammatory activity of their own and other ancillary antiviral activity of their own. So I believe that quite a few doctors, both allopathic and naturopathic and holistic, are now using these agents in early stage patients. They're useless in the hospitalized patients.
Yep.
But in the first 7 days when viruses are replicating, I think they're an important intervention. In the hospital phase, I think we're making big progress.
Yeah, go ahead, Chris.
One of the things when you were talking with Carletta and Shankara a couple of days ago, one of the things you mentioned, Philip, was the fact that if you were a patient, but if you had COVID and you had to go into hospital, if it got to that point, you would know, and I would know, and most of these people on this panel would know what the right thing to do would be, but it wouldn't be done. And that's the biggest frustration. We know now, we've known for over a year, how to treat COVID. We knew early on about corticosteroids. There was a lot of debate about this. Should you use corticosteroids for a viral disease? Well, yes, you should, because at that point, it's not about a viral disease. It's about the reaction of the immune system. You shut it down, full stop. But, now we're a year on, and people still aren't understanding this basic principle that you've got two diseases. You've got a viral disease that is rather similar to flu, and then you've got another phase that goes hyperinflammatory, which is the dangerous bit, of course. But that is still not being understood. And it's the timing. And of course, this has been brought home to us by the work of Shankara and others. And there was a paper that came across when I was rewriting a review on nutrition or pathogenesis, nutrition and COVID. And it was talked about, I think it was September last year, that, looking around day 5 onwards was important. That's where the therapy had to go in. And that's what we've got to make people understand. that is the time point. 5 to 8 days. Get the therapy, and then even if people haven't used prophylaxis, you put the therapy in then, and you will stop people transitioning, because once you've got into the hospital phase and the ICU, you're into complicated therapy. You're losing the battle.
I agree.
You have to treat it early, and you have to treat it at a certain point.
So what you're saying,
that's
Chris,
frustration. If you were in hospital with this disease, you'd say, well, I know what to do with it, but you wouldn't get the attending medical people to use those therapies.
Yeah, before you ask the question, John, I'm interested in everybody's views. So say, Marcus, what is your thought?
In that vein,
No. In terms of where will the world be in a year's time with COVID?
Well, yes, the oral therapies, they need to advance. They're going to start realizing further away we are from the original spike protein designed for these vaccines, the less likely these vaccines are going to work. We're going to start looking at all the therapies and all across the board arsenal of tools that we need. And hopefully, we're going to start recognizing that basic nature of the disease. We need to treat it differently at different stages. But still, I don't see how we can accelerate the intranasal therapies. I don't think hopefully, they're going to gain some, traction in that sense. I can tell in Dominican Republic right now, as part of the national protocol that I was part of, because back a year ago, part of the way that Dr. Shetty was doing work, the 8th day, I published in March last, essentially the same. We use this same protocol in the hospital. The only physician ending on the ICU is the psychiatrist that you don't believe me. But again, I think we're gonna advance in that front, and but my concern is this: we have advances in medicine, we learn how to treat it. How about the poor country? Cannot afford a oral treatment of $400, $500, $600, $700. They cannot afford these therapies. We need to develop and focus on therapies that we can have and share with all the world. And that's one of the most important things that I would like to, advance in all this.
Yeah. And Jennifer, your thought?
Yeah, so when this started January of 2020, being a virologist, I thought one of three things can happen. So we had SARS, the original, in 2002, 2003, and that disappeared just the way it mysteriously appeared. So that was one option. The other option is that this could burn itself out and disappear after some time and be gone. And then the third option is that this virus just sticks around and then becomes endemic. Now, because of the response to the virus and the pandemic, I think we're stuck with option number 3. So I think it's going to be endemic. We're going to have sporadic outbreaks. As from the virus point of view, we know with other viruses, initial introduction to a host can be very pathogenic. So as the virus and the host learn to work with each other, hopefully become more attenuated. So I'm kind of skeptical about all this talk of variants, because the virus is already so well adapted to humans and to transmission that I feel like a lot of these variants and the changes would be more maladaptive for the virus. But in terms of treatment, early on I would get calls from clinicians asking for testing and what to do. Treat your patient. So like you said, Chris, it's very important. What we can do is offer supportive care and treat the symptoms. So that wasn't being done at all. And it wasn't until the patients were getting to the hospital where they were like, oh, now we have to do something. So nip it in the bud, getting treatment. We go to the doctor for, a cold. What do? Treat the cough, give us azithromycin or prednisone, something. You have to give something to the person upon the initial diagnosis. So I think starting there, and I do think that we are learning. So we're learning what works. hopefully physicians are starting to just try things, just do things, try things, treat those symptoms. But I also believe that, what we've talked about here is very important, is looking at overall health. So underlying health is going to be important. And I had a lot of public health nurses working with us. And what we said was, hopefully people will see how important, keeping yourself healthy is. So, using these high dollar therapeutics, when we would see doctors using remdesivir, it's IV, you can only get it in the hospital. We need things that are going to be broadly available to people. We have a lot of health disparity, not just in Hawaii, not just in the US, but across the world. So we need things that are going to be cheap, effective, and also, I think somebody mentioned, have a long shelf life. So something that can be easily shipped, something that can be easily delivered. And I think, that's very important. And it goes beyond SARS, right? It's going to have to go beyond just this virus, because we're going to see more introduction of novel pathogens into the human population. So we need to think about beyond this. Avian influenza is still out there. It's still trying to make a break into the human population. So I think a year from now, I think we're going to be in a much better place. Doctors are learning better about how to handle these infections, and more treatments are coming available.
Okay, then, Marcus.
I have to say bye. I have to jump into a plane now. I'm going to Paris, then Dubai, then potentially India. So it's been a pleasure. Thank you very much, everyone. And we're gonna keep in touch in LinkedIn and the other social media.
Okay, great stuff, Marcus. Thank you.
Nice to meet you, Marcus.
Thank you.
So
Stan, your thoughts?
Yeah, I do see it similar, I think, to what John had said. I think there are a couple interesting things, though. During COVID, myself, I can speak for myself, have gotten more unhealthy. I think that people talk about the COVID-20 or COVID-30, that the pounds they put on during this time. I'm certainly not immune to that. The question becomes, as we get better therapies, the landscape of health in general is going down. So what is the impact of that, over time? I don't know. I mean, maybe we were able to combat these things, but we're also increasing obesity, hypertension, and other things that we know are risk factors. So that's a question mark in my mind with that. I guess a year from now, where I see us, unfortunately, I do believe we're going to be coming into more difficult variants. And as we think about, I live in the United States, so I'm more worried about the U.S. and vaccination and so on. But there are vast parts of this world that have no therapy, nothing to protect them. And unfortunately, we live in a global environment. And guess what? The next very bad variant that pops up somewhere in the world, give it 3 months, it'll be here. So we have to attack this at the global level. And that's what I worry about. We're not doing that.
So before you say anything, John, I'm just curious to hear Debbie's thought. Where are we in a year?
Well, I believe in a year I would have found an investor and some good business partners to help create this medical device at a much better cost point, and we'll have it in every single home because it works. And I've got 100% success so far in COVID, and including anosmia. And so, it works on all mutations. I reckon that trend is going to continue. And I don't feel confident with the work happening in the PC4 labs and the growth of the PC4 labs. I think within a year there's going to be another virus that's going to be leached on us. And maybe it might be 2 years, who knows. So, yeah, I think we do need to have these discussions, and we do need to be better prepared than we currently are.
Excellent. Yes, John. Sorry to interrupt you.
So I'm kind of optimistic. I understand, and Jennifer, there are worse variants that are emerging. They're more infective. So the adaptation on a natural basis does happen. But what we've got here is an artificial situation. We've imposed lockdowns that influence all kinds of, epidemiological trends. We've got, the use of monovalent vaccines, which are incomplete in my view, and some people actually believe they engender or encourage variant, evolution.
Yes. I'm one of those people.
Philip, you had Geert van den Bosch, who's become very controversial, but I think he's got a point. These variants are erupting all over the place now, and in places where there were initial uses of the vaccines.
Yes.
And so maybe the vaccines are engendering something. We just don't know, but certainly they're evading these vaccines, and some of the vaccines are not working as well anymore. I think that the polyvalent vaccines, not necessarily the killed ones like killed whole viruses like the Sinovac, but constructs of codons for all kinds of portions of the virus, not just the spike protein, will be much more protected. I believe that people are now beginning, across the health spectrum, understanding that general health is very important, and that nutritional, nutraceutical, and even pharmaceutical prophylactic interventions, including what we've talked about today, like nasopharyngeal, treatments and sterilizations, are going to be a very important part of the prophylaxis, whether you're vaccinated or not. Okay, and in the meantime, two trends will happen. More and more people will use old therapies like zinc plus hydroxychloroquine or zinc plus ivermectin, and include doxycycline in that mix. So that's that group of people in the front line, of that kind of ilk. And in the meantime, the Mercks of the world are developing their, more traditional, regulatory approved antivirals that will be available to treat early. Like favipiravir is a molecule that's available in the Far East, not available in the West, but it seems to have activity on viral loads quite well. And molnupiravir, the Merck molecule, is an analog of that. So that will come along. If the governments cooperate to make the molnupiravirs available to the third world, to treat early disease, or if the third world, as is happening in India now, starts adopting ivermectin with zinc, as an alternative to all that, a cheap, available alternative to that. As these therapeutic trends emerge, then I think that the hospitalization rate will plummet. And at the same time, we get these polyvalent vaccines coming in. I think that the endemic aspect of this disease is going to arrive far sooner than most people believe, and that we will have an endemic problem with this disease, and it'll become like anything else in the viral world. Some people who are frail, who are, have other comorbidities or very elderly and frail, will succumb as they do to the flu. And we'll have to cope with that.
Can I say just? Mm-hmm.
I agree in terms of the vaccine. I think the spike protein was the wrong way to go. And I think I've posted about this. Using other things like the nucleoprotein and the E are certainly important,
yes.
Getting those in there. I've seen like the inactivated vaccine. I think China has one, but it's not faring very well. So I think,
the inactivation is destroying the epitopes, basically.
Yeah, we cannot rely on vaccination. That's just, everyone thought that was going to be the magic bullet. Therapeutics with viral infection is really the way to go.
I think so. I think it's a very important way to go.
Yes.
Can I say one important point that I think that I found from my research point of view keeps on getting missed, is that the primary mechanism of this virus, and I guess if it changes over time, was that it evades the interferon response in the first 3 days. This is why you don't get a runny nose, you don't get a temperature. It hits the lungs before your body even knows it's there. And so this characteristic is part of the reason why I've been interested in the upper airway solutions. Because if you can't know when you are infected, you do have to use some kind of prophylactic system in order to reduce that risk, because it is the weakest point. And I still do believe that the future and this is why I'm sorry Vanya wasn't here, is the future is going to be with nasal vaccination. If you get mucosal vaccination, you know, everything else is irrelevant because the virus can't replicate and so it can't get in the body. So your target.
Yeah, a little bit with rhinovirus. So there was a paper that came out that people had been infected with rhinovirus were protected from infection with SARS-CoV-2. So that goes to the stimulation of the interferon in the nasal passages, and then that preventing the infection cycle. So, you know, maybe we just infect everyone with rhinovirus.
Yeah. Yes. Yeah. Get a nasal spray. Give everybody a cold.
Coronavirus. Why stop at give them non-noxious coronaviruses?
Yeah.
And so you get some cross-reactive mucosal antibodies right away. You know there were some early papers, for example, that were looking at BCG and MMR vaccines as being protective against COVID as well. I think those were non-specific immune stimulant effects, you know, rather than any specific cross-reactivity. But they're all, look, you know, the interesting thing about this pandemic is that it engendered such fear, an inordinate fear, you know, compared to others that we've had that are much worse in their portion, you know, in terms of mortality and morbidity. But you know, when things started happening in Wuhan and they showed pictures of people keeling over in the streets, and when the Wuhan city government shut down everything on the pain of actually being shot if you came out of your apartment, that I think instilled this massive panic that went through the world. Now, the good part of all of that is that it's being engendered a tremendous amount of research on all kinds of approaches to this thing, be they antiviral, be they immunologic, be they nutritional, be they physical, like what Debbie's doing, etc. And you know, there are knock-on benefits from all of that in terms of other preparations for the next time around. I mean, let's go back to what Philip had said in the beginning with his own research and his suspicion of the autoimmune effects of this thing. That happens with other viruses, right? I mean, there's post-flu syndromes. There's, you know, after the 1918 pandemic of influenza, 10 years later, there was an enormous increase in Parkinson's disease. And you know, Parkinson's disease is a neuroautoimmunity, in my view. We don't know yet now, at this point, what these long-term sequelae of this virus is, as opposed to normal coronaviruses. Nor do we know what the long-term sequelae of these codon-based vaccines are. Really, they just haven't been done. That couldn't be done, and it's a leap of faith that there will be no long-term sequelae. For example, Philip, let me pose this question to you about vaccines that are being used now. The vaccines that are being used now are engendering spike protein. Your hypothesis says that spike protein anneals to ACE2, correct? And especially serum ACE2, which is floating ACE2 where it shouldn't be. You know, ACE2 should be on the membrane of cells, right?
Yeah.
But it's clipped off in certain conditions like diabetes and hypertension and obesity, metabolic syndrome, all of those things, which are surprisingly the vulnerable groups of comorbidities in COVID. So, you know, there's an analogy there that holds. But wait a minute. If vaccines are increasing spike protein and people have ACE2 in their blood, will the vaccines not induce the autoimmunity which you're invoking?
Well, this is part of the question, you know, and this should have been one of the first questions that was asked, because we do know that the serum ACE2 binds to the viral spike protein. So unless you formulated the spike protein in such a way that it wouldn't interact with the serum ACE2, then yes, absolutely. And when you look at this is the big concern, when you look at the presentations of people, they're usually presenting with side effects, or if they're going to have any, sometimes about 2 weeks after. And that's usually about the time that you have antibodies being produced. In certainly the IgG would start to peak by that point. And so there is concern that the spike protein itself could cause damage, but I would suspect that if it was going to do it, it would do it within a couple of days and we would see issues. But for the vast majority of people, it's say about 10 days, 2 to 3 weeks after, if they're going to have any problems, that it happens. And that suggests to me more of an immune-mediated mechanism.
I've got a slightly, sorry, Philip, I've got a slightly different view on this, and I'm not denying that hypersensitivity comes into this, but I think there must be some kind of cell death event occurring at a certain time. Whether this, how widely this is occurring, I don't know, but I would suspect that there's a release of damage activation PAMPs at a certain time, which is kicking off, perhaps via some kind of receptor that is kicking off what could be a sort of hypersensitivity response, a histamine release, the activation of other systems, the production of interleukin-6, because interleukin-6 is taking off again. And just going back to what I was saying earlier, we haven't got good data. What we need is good data for the early phase. We need to know what's happening with the cytokines in the early phase. The only data I can find is going back to flu, and there's not an awful lot of data there either. So we're missing a huge chunk of data as to why are people ill? Why do people get symptoms? What makes you ill?
So I can tell you from my work with influenza. So when I published a few papers on this, and we took a non-lethal virus, and when you deliver it intranasally, you know, in mice, you may get a response. The mice do just fine, no sickness, anything. But now if I aerosolize the virus and the virus gets into the lungs, what I saw was this massive damage in the lungs.
Yeah.
I mean, within 3 days, the mice are dying. So this is a non-lethal virus, but the response is driven by IL-6 and also reactive oxygen species. So neutrophils flood in, reactive oxygen species, and then you get this inflammatory response. So in my work is what I showed is it's the immune response. And where the virus is getting localized, that is the reason for the damage. And I think it's similar in this case with the SARS.
But why do we have situations where you have some individuals that have a very moderate initial response and then they transition? So this is quite difficult to answer.
This is a bit that I've been focused on. So we are now doing a third paper looking at what we call the molecular basis of the autoimmune response. So I'd spoken with Dr. Shetty, and he mentioned the impact of histamine. I knew, therefore, that at the day 11, the mast cells were getting activated. The question is, what was the mechanism? So based on what we're looking at is that I think that the immune response, just like what you said, Jennifer, with the virus hitting the lungs, when you look at even children and people who are apparently asymptomatic or subclinical, up to 70% have quite a significant lung inflammation. Just because they don't have any symptoms doesn't mean there's nothing going on. That's the first mistake. So what it looks as though it happens is that because the virus has evaded interferon, hits the bloodstream and the body overreacts, that's the phase that we have the risk of an autoimmune or hypersensitivity response occurring. Now, this is part of why we want to research, and my research is focused on ACE2, but I agree that it could be on another protein or another mechanism that's doing it. But there is something there at that point, because the pattern is so consistent, timing with IgG production, that there is something that is happening that is creating this very, very powerful immune response in a consistent pattern.
Right. So if you were to hypothesize, as you have, Philip, that there is an agglomeration of serum spike a serum ACE2 onto the virus, right? Onto
Yes.
the protein of the virus. That forms what is a pseudo-immune complex.
Yeah.
Now add to that perhaps coronavirus antibodies from past coronavirus exposure.
Yes.
Yes.
And now you've got a furball of extreme complexity as kind of a novel immune complex of a mixture of ACE2 together with corona antibodies. That could engender a macrophage reaction that is quite unusual. And that can then lead to mast cell degranulation, histamine release, eosinophilic response. I don't know whether or not there is, this is a question to ask, in biopsies of fatalities, have they shown any eosinophilic invasion or eosinophilia?
And this is part of the reason why, even though there is a histamine response, I'm not going towards it being eosinophilic. Although there is a response with regards to eosinophilia being almost zero in a lot of cases, something is happening to them.
Yes, there is.
So something is happening.
But an immediate extreme.
Yeah, but they're not.
So you get an eosinophilopenia, I guess.
Yeah, but it's not accumulating in the lungs. This is the thing, is that so I think say for instance when I've spoken with Dr. Shetty and he's speaking about almost that eosinophilic pulmonary infiltrate, it hasn't been borne out with the biopsies, but the mechanism, something looks very similar about it. You know,
and some people, you know, I mean, again, this is I have an interest in nutraceutical, you know, pharmacology. I'm just crazy that way. Some people have used extremely high doses of vitamin C in intervention in the hyperinflammatory condition, and we know that a very high dose of vitamin C does stabilize mast cells. And they reported some salutary benefit from using it. Could be through that mast cell, you know, stabilization, similar to the antihistamines, you know, negating the mast cell degranulation effects and histamine release.
Yeah, so that goes along with vitamin D. So I had a grant funded to look at the effects of vitamin D on replication of influenza in lung epithelial cells, because we know that the vitamin D receptor can get stimulated, and we saw a decrease in virus titer. And, you know, vitamin D plays a big role in T cell development. So this is where I love seeing a lot of this research and people talking about vitamin D3, because there is a very significant role in immunity and memory for virus infections. So vitamin D being key, which is why the lockdowns and shuttering people in their houses made no sense to me. Like, get them outside in the sun, you know, especially here in Hawaii. Just get out to the beach, get people outside, get them, you know, stimulate that vitamin D, because so many adults are, and people in general, are just deficient in vitamin D. So I think that's key too. Yeah. And, you know, I take high dose vitamin D every day.
Now listen, guys, I think we could continue talking all evening, but we are going over 2.5 hours. This is tremendous. And it's been an absolutely fascinating conversation. So listen, I think that we should wrap up now. I think that this is probably a whole conversation to have on its own. So we'll probably think about how to take that up. Again, thank you very much. We still have 75 people actively listening to this conversation afterwards. So it's clearly very, very interesting to a lot of people. So great. So thank you very much again, everybody. Have a great morning, evening, wherever you are. And I'm sure we're going to speak again soon.
Go to sleep,
Take care, everyone.
Go to sleep,
Debbie. Thank you for sharing. I learned a lot. So
yes.
Have a good day.
Stay in touch with each other. Why don't you?
Yes, certainly.
Love to. All right.
Okay, then, guys. Okay,
Bye-bye now.
Bye-bye.
Bye.